All right. All right. Oh, sorry about getting the quiz up so late. I was going to get it up Wednesday night, and I went to sleep. So, yeah, that's what happened. So I got it up last night. All right, cell membranes. So the first four questions. These four questions we're going to move through pretty quickly, okay? These are the portions of what happened on Wednesday that probably were like, okay, I got this. And then it's like you hit something else, and you're like, well, I no longer have this. I don't know what happened. Okay, so what are the lipid components of the cell membrane? How do other biomolecules function in membranes? What is the structure of membranes? And how does the membrane function? Okay, again, these we're going to move through fairly quickly. Okay, buckle up. Let's do it. So what are the lipid components of the cell membrane? Okay, you know this. Cell membranes are made primarily of phospholipids and sterols. Okay, phospholipids and sterols, and you know that there are different types of phospholipids, but you also know that phospholipids are polar and nonpolar. That there's this really complex end that's got a phosphate group and some other really polar functional groups that make for a polar end of this phospholipid. And then we've got a nonpolar end with two fatty acid tails. Okay, and you know that some fatty acids would be much better to keep something from solidifying than others. What types of fatty acids are often solidify at room temperature? Saturated, right? And you know now that a solid membrane is not good, right? A solid membrane is not good, so that means you should be able to infer that the fatty acid tails on these phospholipids are not saturated, they are rather what? Unsaturated, okay? Which prevent them from solidifying at room temperature. Okay, phospholipids are both hydrophobic, their polar end associates very well and very effectively with water. Their nonpolar fatty acid tails associate very poorly with water, but associate very well with nonpolar materials, like other fatty acid tails from other phospholipids. You also know after Wednesday that phospholipids are self-assembling. Now, I know Dr. Francis told you most of these will form micelles with a very, just very simple and even almost like a mirror image. The inner and outer layers are a mirror image. It's almost entirely phospholipids, which aren't the kind of, it's not a plasma membrane, but the phospholipids will form these membranes spontaneously when you put them in water. And they get semi-solid at low temperatures. Even with those unsaturated tails that have all those bends and twists, they do. If all you have is phospholipids, like you form one of these micelles, at lower temperatures, they actually will begin to solidify. And that is not good because a solid structure becomes brittle. And brittle structures do not make effective barriers, right? And they don't facilitate great compartmentalization, which those are the key things that the cell membrane needs to provide, right? You know that. You know that. All right, so sterols. They are also polar and nonpolar. Now, the particular chemistry involved is a little bit different, but we still end up with a polar functional group. What functional group is this? This is a hydroxyl group. So we've got a polar hydroxyl group, and then we've got this big carbon ring forming this nonpolar entity. So they're still polar and nonpolar, meaning like phospholipids, they're hydrophilic and hydrophobic. And it also means that they're going to be arranged similar in these phospholipid bilayers. That the polar part will stick out where that big polar head is of the phospholipid, and the nonpolar ring will be buried within the membrane like the fatty acid tails. Okay? Make sense? Y'all saw this, right? A lot of this on Wednesday. Now, in animal cells, these sterols are mostly cholesterol. Cholesterol is a sterol you're familiar with, probably most often used negatively, right? Like you went to the doctor and you had a physical and they're like, oh, your cholesterol is a little high. You're like, well, I eat a lot of bacon, so it kind of makes sense. Not really giving that up. So, you got to figure something out, right? Did you ever talk to your doctor that way? So cholesterols are very healthy materials, right? There's good cholesterol, there's bad cholesterol. Anyways, it's another story for another time. But the cholesterols in animal cells do a wonderful job of keeping the membranes from becoming solid at low temperatures and keep them from falling apart at high temperatures. Okay? So these cholesterols, they're stabilizers. Again, they do a really nice job of stabilizing the membrane, maintaining that fluidity. Because you want these membranes to be fluid. And when you hear fluid, you can't just think water. That is only one type of fluid. Okay? Fluid is just anything that's not solid. Okay? Liquids are fluid, gases are fluid, even gels are fluid. Like Jell-O, right? I can't say Jell-O without doing this, because it's just, I mean, that's what it does. It does that all the time. It's like you don't even have to shake it. It's just doing that, right? You set up a camera in your fridge, and you just, have you ever done this before, and have it look at the Jell-O? And it's like nobody's even opened the door, and it's still just in there jiggling. Man, that is an active material there. Anyways, sorry. So here's a phospholipid. Got the big old polar head, these non-polar hydrophobic tails. And again, a lot of these, you're going to see a lot of these double bonds, creating these unsaturated fatty acids with these kinks that keep them from getting too tightly concentrated and solidifying. You don't want a solid membrane. And here's a good example of a fluid bilayer where these fatty acid tails, I mean, these phospholipids, they're rotating around. They're even moving around this membrane like millions of times every second, moving, changing position on this layer. Now then you have machines that will move them over to the other ones. Did Dr. Francis tell you about some of these? Did he tell you about flipase? Yeah. That's just a wonderful name, but it's very fluid. These phospholipids move around. This is what a frozen membrane looks like. Very rigid, right? There's no longer a dynamic nature to these fatty acid tails. The lipids aren't moving around or flipping layers, and it's very brittle, very brittle. And brittle things, again, do not make great barriers, and they do not do a good job of compartmentalizing various areas. Man, I don't know why I'm struggling to say that. So here is cholesterol suspended in this phospholipid bilayer. Here's that polar functional group, that hydroxyl group, and then here are these complex carbon rings, very nonpolar. And again, it's in there to just, it keeps these phospholipids from getting too tightly packed and solidifying. But it also helps to stabilize them at high temperatures and keeps them from falling apart. Okay? Make sense? It's good stuff. It's good stuff. All right, so how do other biomolecules function in membranes? How do other biomolecules function in membranes? And again, you talked a little bit about this in a lot of detail, but it's good stuff. So first we'll look at proteins. We've seen these before. You know that there are proteins in cell membranes. We've talked about this before, that are receptors, are transporters, and are labels. And so here we've just kind of taken that a bit to another level. We've added one additional thing, and then these, the identifiers, labels, the same thing. So we've got transporters, and why are there transporters? To transport materials. And you're like, okay, cool, but why do you need to actually have proteins to transport materials? Why can't materials just move across this fluid membrane? Some things don't move well across that membrane. What types of things don't move well across the membrane? Things that are polar, because they'd have to interact with that big old non-polar environment, and things that are really large. Like sugar is polar and large. Okay? You're like, well, what about glucose? That's just one sugar. That's true, but it's super polar. It does not move well across. So we got transporters to do that. Identifiers, again, to declare self. We've seen that before. Receptors to bind material in the extracellular environment. And then there's one more. These are a special type of receptor that are adhesion molecules that attach the protein to other cells. We saw that already. Or to attach the cell to the extracellular matrix, which we've seen already. No, it's fine. So then we've got sugars, and the sugars in cell membranes are attached to either proteins, and so we'll call them glycoproteins, or they're attached to the lipids. Phosphatidylinositol is oftentimes has a sugar attached to it, right? And you all remember phosphatidylinositol, and you remember that G-coupling protein receptor triggers converting phosphatidylinositol into a different type of lipid, which then that inositol group will break off as IP3, which will then go and act as a transcription factor. Cell signaling, right? It's all wonderful stuff. Anyways, we've got these sugars, and sugars are sticky. And so when you see sugar, you need to be thinking that these are oftentimes going to be associated with cell-to-cell adhesion. Okay? Cell-to-cell adhesion, or at least giving another label to the outer surface of that cell. Like in red blood cells, the A and B, right? You're familiar with A blood type, B blood type, and then O. They correspond to very specific sugars on the surface of the red blood cell. Yeah. Yeah.
Is it the polarity that makes these things?
Yeah. I mean, and just the distribution of the polarity. Yeah, the nature of the bonds within it. Yeah, there are a lot of polar bonds in sugars, and they're kind of distributed all around the molecule. Yeah. This is a good question. All right. Any questions? Okay, so here's a typical transporter. And so this is an ion channel. And so it's facilitating the diffusion of what? Of ions. And because ions, like everything else, they're going to move down their concentration gradient, right? We saw this in lab yesterday if you had the Thursday lab, but on Monday, if you had the Monday lab, things move down their concentration gradient. But some things are too big or too polar to move through the membrane, like sucrose. But they can move through if you've got a specific channel for them. But this can allow ions to move down their concentration gradient. And so we saw this already. We call this a transmembrane protein because it moves all the way through both layers of that phospholipid bilayer, moves through the membrane, transmembrane protein. And if you look at the amino acids on the outside of this protein, what would you predict that their R group would be? Polar or nonpolar? On the outside?
Polar.
Outside, not on the inside. The inside's where the ion's moving, on the outside.
No.
The outside's exposed to these lipids, right? Those R groups are nonpolar. But the R groups on the amino acids on the inside are polar. And that's what allows these ions to associate with them and to move through. And then so this is what that channel would actually look like, sort of. It's like a cartoon version of it. And then here's a simplified model of what these transporters look like. Okay. So again, polar R groups on the inside, nonpolar R groups on the outside. Okay. All right. Let's do one more and then we'll do a lecture break. I know we're pushing hard, but you can take it. You can take it. Just get to the point right before you snap. But then let you go and you'll rebound back. It'll be good. All right. And then you'll be able to stretch further next time, you know? All right. So what is the structure of membranes? What is the structure of membranes? So I want you to know, I don't think I've mentioned this before, but a lot of what you learn in cell biology, it's like this is the dominant theory of how it works, right? And it seems to have a lot of data that support it, but we may figure out it's entirely wrong, which is kind of fun. You know, like 10 years from now, they might learn something completely different than I did. That's why it's cool, you know? It's like math. You can help 10 years after you take algebra, you can help somebody through algebra, right? Algebra doesn't change. Cell biology, it changes. You got to keep up with. Anyway, sorry. So the prevailing model of cell membranes is what's called the fluid mosaic model. That it's a fluid structure in which phospholipids can move freely and fluidly. You like that adverb, right? The fluid mosaic model, fluidly. And so phospholipids can move around freely within the layers. Unsaturated fatty acids, we talked about, maintain fluidity at low temperatures. Cholesterols reduce fluidity at high temperatures. Okay? And so the idea here is, again, this is a very fluid structure. It is obviously not solid. Obviously not solid. It's too fluid for that. You know what's kind of cool? is glass is not a solid. It's not a liquid, but it's not a solid. It's too fluid to be a solid. So what that means is that glass is subject to move just like other fluid structures are. But since it's not fully liquid, it has a really, really, is it low viscosity or high viscosity? Viscosity is the tendency to move, tendency to flow, so it's a low viscosity. But if you go into like a really old house, if you go into a house, we don't have a lot of these in Southern California because earthquakes, chew up old houses like tornadoes chew up trailers, right? And so, but if you go into like a really old house, well, if you had a tornado destroy your home, I'm really sorry. Okay, but if you go into a really old house and you look at the windows, they're thicker at the bottom than they are at the top, and you can see that fluidity of glass, and it's kind of cool. Sorry. It's like a plasma membrane, where it's not a liquid, but it's definitely not a solid, somewhere in between. Right? It shows you that those states are not discrete types of existence. It's really a continuum, right? Where you've got solid on one end, gas on the other, and then you've got weird, crazy things like stars. What are stars? And it's like, oh, it's this weird plasma. But anyways, you've got this continuum, and it's like a membrane and glass are somewhere in between a solid and a liquid. They're too fluid to be a solid, but they're too solid to be a liquid. Okay, does that make sense? Okay, but then the mosaic is we've got all these other materials that are suspended within that fluid that is the lipid bilayer. Now the protein's mostly floating individually, although some of them are anchored in place by the cytoskeleton. Because if you want something to keep from moving around, you've got to anchor it in place. And you've got this massive cytoskeleton under this fluid membrane that does a wonderful job anchoring things in place, right? This is what microtubules do. They anchor organelles in place. They provide roadways for vesicles, It's just a great thing to attach a protein to, if you want the protein to stay put. Now, integral proteins, they're embedded in this membrane. So a lot of times they're just as fluid as the phospholipids. But the peripheral proteins, they're like attached to the outside of the membrane, usually through hydrogen bonds or even van der Waals forces, even weaker bonds. But what it does is it creates an asymmetry in the layers of the membrane, where the outer membrane has different proteins than the inner membrane. Okay? Well, the inner membrane than the inner layer. You're like, there are two membranes? No, there's only one membrane. The outer layer has different proteins than the inner layer does. Creates this asymmetry. This asymmetry. Okay. Any questions here? I know we're kind of boogieing through here, but we'll slow down in a minute, like at the end of the semester. I mean, isn't it encouraging to know that you're going to have a quiz for every single class period we have except for when we do an exam? So it's just consistency, right? It's almost as like, knowing that the sun is going to rise every day. I mean, it's right up there with it, right? Or right up there that if you own a dog, like your dog's going to be happy to see you. My dog hates me. Like, absolutely despises me. And so I was gone, right, which is why Dr. Francis taught our class on Wednesday. And I would talk to my wife every night, and she'd be like, man, the dog, he's loving life. Like, he's running around the house freely. He's eating his food without cowering in the corner out of fear. And then I get home, and man, he's miserable again. He's cowering in the corner. I don't even know why. I'm so nice to him. Anyways, sorry. It's just, I'm working it out, right? It's a hard issue. All right. So here are these proteins that are suspended. Here are some integral proteins, again, spanning the entire phospholipid bilayer. And then here's some peripheral proteins that are attached to only one side. And again, they are attached by typically very weak bonds. Hydrogen bonds, van der Waals forces. But even with a weak bond, if you get millions of them, they can be attached pretty firmly. Like this is like a G protein. Coming back to what you learned on Wednesday. These G proteins are peripheral proteins, but the G protein coupled receptor that they work with is an integral protein. It's all coming back, right? All right, so here you can see the fluidity even of proteins. Here they took 2 cells. So they took a human cell with human proteins, and they fused just the membrane of it with a mouse cell membrane. And so initially you get all the mouse protein on one side and all the human protein on the other. But then after 40 minutes, they're all mixed and distributed. Because it's showing you even those integral proteins are fluid. They move around with the phospholipids, unless what is true? The membrane's frozen. That's cheating, right? It's like, but wow, this sandwich is always going to taste delicious unless what is true. And you're like, unless somebody ate it before I got to taste it. I mean, yes. But unless they're anchored in place, right? Unless they're anchored to the cytoskeleton. And then it prevents them from moving around with the fluid. Sorry I stuttered there. I got really excited. And so here's the experimental research that was done to show the asymmetry in the membrane. So they froze the membrane, they solidified it, and then they pried the 2 layers apart, and each layer is different, has different proteins in it. It's asymmetrical. And this is what Dr. Francis was talking about with this compartmentalization. That yes, a big role of the plasma membrane is a barrier, but maybe even a bigger role is compartmentalization, to create a different environment inside the cell than outside the cell, so that the chemistry is different, so that the reactions that happen are different, so that life works. That is good stuff. All right? So here's what I want you to do for our lecture break. We're going to take about 2 minutes. You're going to work with those around you, and you are going to come up with several different types of integral proteins. So these aren't proteins that are peripheral. These are proteins that are going to span through the entire membrane. Okay, and you know they're going to have to fall into basically one of 4 categories now, right? Our receptors going to be integral, right? Our labels going to be integral. Our transporters going to be integral. Our attachment protein's going to be integral. Okay? You probably don't need 2 minutes. How about 1 minute? Is that enough time? I think it is. 60 seconds. Starting, and I want you to come up with 3 different integral proteins. And try to get it specific, not just like, oh, cool, like of these 4, let's choose 3 of them. Because the answer is there actually are 3 of the 4 that will have integral protein. But like very specific types, okay? 60 seconds, starting out.
What did he have?
Are they the one that's sticking through both layers?
All right. Hey, your minute's up. Stop talking. It's not a joke. Gosh. I think it's hard when you have a professor that makes a joke out of a lot of things. It's hard to even take me seriously, right? I think I have the same problem with my kids. They're like, are you joking or do I really need to clean my room? Like, have I ever once told you a joke about cleaning your room? It's not funny. Your room looks like 5 homeless people stayed a night in your room. There's nothing funny about that. They're like, that's a joke right there. I'm really confused. Okay. So what are some very specific integral proteins? Yeah, Tara. Aquaporins. Aquaporins are transporter proteins, and they help water get through the membrane. Now, we know water's polar, right? I told you water is about as polar as a molecule can get. But water is small enough that it can diffuse across membranes. But if you really want to move a lot of water, you put aquaporins in the membrane that help it to move more efficiently and more quickly. And you're like, why would cells want to move water a lot across their membranes? That's a wonderful question. There are cells that have to regulate the thickness of the extracellular fluid, and they oftentimes have to pump out massive amounts of water to thin out that extracellular fluid. Can you think of any cells that might need to do that, where you've got a fluid that can get too thick in our bodies? Okay, so blood, maybe lining the blood vessels where you've got to have a lot of movement of water. Inside the lungs. Okay, inside of the lungs, you've got a thick fluid there that can trap materials so that they don't penetrate your tissues, and that can get too thick. And so you've got to be able to move water efficiently, because if it gets too thick, then it's very difficult to exchange gases. It's not good. It's not good. All right, any other specific integral proteins? I mean, you saw one on Wednesday. That G protein coupled receptor. Right, that bound some ligand and triggered G protein to convert phosphatidylinositol into a different chemical type of phospholipid that loses that IP3. What is it? An inositol triphosphate is what it loses, right? So they're receptors. In that case, a G protein coupled receptor. That's a very specific integral protein. Yeah, Tara. Voltage gate proton pumps. Or the channels, okay. Yeah, so you've got so you've got ion channels, right? That will let ions in. But you can probably imagine you don't want those open all the time, right? Because then ions are just flowing across all the time, and it's hard to control the charge of the cell. And so there are what are called voltage-gated ion channels that are only triggered in the presence of an electrical signal. So there's an electrical signal that says open up the channel. And then massive amounts of charged materials move. And when you have charged materials move, what happens? Just think about when you plug something into the wall and you have massive amount of charged materials moving into your cord, what happens? It powers your device, right? You've got enormous electrical potential happening. Okay, and that's how a lot of cell signaling works, but we'll get to there in our last week. Good. All right, yeah.
Is that like nerves?
Yes, the impulse, the action potential. Yeah, usually it's an initial chemical signal that opens up maybe one channel and creates enough destability that it opens up all of these channels. Sodium ions run in. You get this massive change in electric potential. You get this electricity that runs down the membrane of the cell and then triggers at the end a release of the chemical that gets the next neuron going. And then ultimately it gets to a muscle, and it does that in your muscle cell, and it contracts your muscle, right? And then if you've got botulinum toxin in there, your muscles can't relax, and so they just stay contracted forever. Sorry. All right, so of those 3 types, the only one that are typically not integral proteins are labels. Labels tend to be peripheral proteins and peripheral glycoproteins. But other than that, attachment, receptors, transporters, those are almost always integral, spanning through the membrane. All right? Make sense? Cool. Happy Friday, by the way. I don't think I've said that yet. This is a great Friday. I believe it's February 8th. And you know what that means? I have no idea what that means. Well, it's almost Valentine's Day, and that's pretty cool. It's almost my father's birthday. That's pretty cool. Is it really? Is your dad's birthday February 11th?
No, it's February 10th.
Is it really? That's my dad's birthday. They might be twins. What if your dad is my dad's twin? And they didn't even know. Is he? My dad's not. My mom's a redhead, though. Yeah. And I bet my dad's probably older than your dad, just based on statistics. Yeah. All right. All right. How does the membrane function? How does the membrane function? I'm glad you asked. Man, there's a lot of things that have happened in this class today that in no way prepared you to be a better cell biologist. But maybe a better human being. All right. So membranes, we know that they form barriers, they form boundaries, and part of this again is yeah, I mean, it's just being a wall, but a lot of this is to create this compartmentalization where you get different environments. Because just putting up a fence doesn't create a different environment. It might help to facilitate maintaining different environments, right? Like just putting up a fence doesn't mean all of a sudden your garden's going to grow stuff, right? You still got to plant it. The fence may keep the rabbits out until they burrow under it. And then the dynamite comes in. Oh, man. Oh, yeah, so membranes are permeable. They're what we call selectively permeable. Some materials get through, other materials do not. It's like the TSA screening of your baggage. Some of the material makes it through, some of the material doesn't. Jack, when you get to your final destination, you open up your suitcase, there's like a little card from TSA, and they're like, thank you so much for allowing us to rifle through your luggage. By the way, we took everything in there that you weren't allowed to have. Sorry. Have a good day. No? This has not been your experience? Oh, so they can be enzymatic. So some of the proteins that are on these can actually be enzymes and to facilitate reactions. Not like the G-coupled protein receptor that you saw on Wednesday, because what was actually enzymatic was that G protein coupled to it. But some of our receptors are enzymatic. They don't need a G protein coupled to it. They facilitate reactions. One of those types of receptors are called receptor tyrosine kinases. We'll get there. You're going to love them. They're so good. They regulate the transport of specific ions. And we've got a specific type of ion channel called CFTR, which stands for cystic fibrosis transmembrane conductance regulator. I don't know where the C went in the acronym, but it's not there. And this allows chlorine to diffuse out of the cell when the channel opens. And that chlorine diffusing out of the cell messes with the osmoregulation. And so water will tend to follow chlorine. Okay, when chlorine diffuses out, it'll raise the overall solute outside of the cell relative to inside of the cell. You all know now from lab, water is going to run to where there's more total solute, right? So if you allow chlorine to diffuse out but no other ions, you get more solute outside of the cell than's inside, the water's going to flow out. And this, again, water can flow out to thin out that fluid. And this is one of the reasons, one of the pathologies of cystic fibrosis is what? Super thick lung fluid, which often leads to lung infections. It makes breathing really difficult, makes it to where people with cystic fibrosis, there's no cure for it. They end up having to get a lung transplant at some point. Okay, when enough damage has been done that things don't work. Membranes can also bind substances and then transport them across the membrane, like glucose. We've got a glucose transporter that's exactly what it does. It binds glucose and transports it into, allows it to diffuse through the membrane. Membranes can recognize specific molecules. What protein type's going to do that of our four? The identifiers, or the identifiers oftentimes are labels, but recognizing specific molecules, these are receptors that bind very specific molecules, like the G protein coupled receptor you saw on Wednesday. And wasn't it cool, the analogy that he gave you? It's like something bound to the wall of your house and then the wall physically changed to provide the signal. That is so cool. That is so, so cool. Conduct signals with electrical properties. Tara, you brought this up with the voltage-gated ion channels. Oh, man. facilitates cell adhesion and cell-to-cell communication. Somebody define communication for me. I want you to give me a general enough definition of communication that it will work for all types of communication. Davies.
Exchange of information.
Exchange of information. I like it. I like it. Any other ideas? And so yours is actually shorter than mine. I like to say it's the transmission and reception of signals. It's the transmission and the reception of signals. But the transfer of information is good. And so cell-to-cell communication, it can be a chemical signal. It can be an electrical signal. It can be a tactile signal, right? One cell touching another cell. Probably can't be a visual signal because I don't think cells do very well with visual cues or auditory cues. But, I mean, you never know. You never know. You're like, well, what about the cells in our retina? I mean, they actually do pretty well absorbing light. So I guess maybe we do have to call that a visual signal. I don't know. But you know, your eyes don't actually see. Your brain sees. Here's the cystic fibrosis transmembrane conductance regulator, the CFTR. And when it opens up, chloride runs out. Chloride is diffusing. It's moving down its concentration gradient. Meaning a typical cell does what with chlorine? Keeps a lot of it inside of that cell. You're like, where to get that chlorine? It's an excellent question. From what you eat. Yeah. Yeah, salt. I mean, who doesn't salt their food, you know? You're like maybe somebody with kidney problems, but that's another story. Gosh, man, I'm just getting really personal here. Oh, Mia's not even here. Ooh, Mia didn't come from genetics. Anyways, in genetics today, I said something so mean. And I made fun of everybody collectively. So it wasn't like towards a certain person. That way everybody can feel equally made fun of. But anyways, you can ask somebody what I said. Well, I don't even remember the context of why I said it. But I was talking about like all of the important things we have to do in our life, and I remember the context now. So I was talking, it actually works really well for this because, you know, like, after a receptor, I mean, you saw this on Wednesday, like a receptor binds a specific ligand, and then it transduces that signal. And the transduction in that G protein coupled receptor was to convert phosphatidylinositol into a different chemical form that releases inositol triphosphate, right? That's the transduction. So what I did was I created an analogy for this, and I was like, okay, step one is you receive the signal. Step two is you let everyone know about it, right? And that's the transduction. And I said it's kind of like when you want to let everyone know about the negative experience you had at a restaurant, so you put it out on social media. But I was like, I want you to know when I say everyone, I don't really mean everyone. I just kind of mean like you let who needs to know. And I said like you, because you've only got 7 followers in your social media. So really it's not everyone, but those 7 people that follow you, they know all about your negative experience. So I think I hurt some people's feelings. I'm like, listen, it's no big deal. Like 7 followers, that's a lot, you know? All right. Next set of questions. How do materials move across the membrane? Which specific proteins function to transport materials? Why is osmosis so special? Now you're checking how many followers you have? Look what I created. I got more than 7, Dr. Engel. I've got at least 8. How can we predict when and where osmosis will occur? When and where osmosis will occur. All right. How do materials move across the membrane? Okay, we've mentioned this several times. Materials naturally move down their concentration gradient. That's the way materials tend to move. Whether it's just moving through air down the concentration gradient, or it's moving across the barrier down their concentration gradient. That's where things tend to move. That's why if you light or you let off a stink bomb, that smell doesn't linger forever because the smell molecules move down their concentration gradient. So everyone gets to experience it, but eventually they become so lowly concentrated that they don't even trigger anything anymore. All right, sorry. So passive transport, these are any things that don't require net energy input. Passive transport, anything that doesn't require net energy input. And anything moving down its concentration gradient is not going to require net energy input. You're like, Dr. Engel, what if you have to open up an ion channel? That's true. You may have to open up an ion channel, but then that's going to allow the things to move, and them moving is actually going to provide energy in the form of kinetic energy. And you're like, oh, it's so cool. Like, it makes sense. Makes sense. It's like, have any of you worked on a farm? Any of you, if you haven't, you can imagine this, right? You got a whole bunch of pigs, you put them away for the night in their little pen, and then you open up the door to let them go and graze, and man, those things come flying out, right? And you're like, that's a lot of kinetic energy. If you want to use it to do something, you totally can. You ever put like a penny on a railroad track and let a train run it over and it flattens the penny? You got to do that on the ground and let the pigs run it over. It doesn't flatten it quite as much, but it's impressive if you can get like a couple dozen pigs to step on it to see how much you can flatten that penny. Of course, you have to have a hard ground. If it's muddy, then it's like, it doesn't even work. Sorry. Oh, man. I don't know why that illustration came into my mind. Probably because I was thinking about bacon, because we talked about cholesterol earlier, and bacon, I haven't forgotten about it. Man, I watched this cooking show, and they cooked something with duck bacon. And I was like, man, that sounds intriguing. Turkey bacon is awful, right? It's like, to call that bacon is silly. But man, duck, like, that's a fatty meat. Duck's probably just as fatty as pork is. I bet that'd be delicious. But anyways, sorry. I just wanted to share that with you as well. So this is called diffusion, the movement of materials down their concentration gradient. Down their concentration gradient. Oh, man. Sorry, it's right before lunch. I can't shake it. And if it's here, I got to share it. Yeah, so this concentration gradient that a cell can set up, it's a form of potential energy. Because if you allow those things to move, now you've got kinetic energy. And again, that kinetic energy you can use to fuel processes. Now, diffusion can be both simple, in which it's just moving directly through the membrane, or it can be facilitated, where it's moving through what? A protein channel, okay? A protein channel, or a transporter, whatever you want to call it. Now, that works well for materials that are going to move in the direction they want to move. But what if you want to set up the concentration gradient? Right? If you want to set up a concentration gradient, you need more of something on one side of the membrane than there is on the other. Now you have to force materials to move against their concentration gradient, like the salmon swimming upstream, jumping up the waterfalls, right? You've seen that? Right into the mouth of a wading bear. It's like, man, they worked really hard. And that's why when they get up there and they lay their eggs, they die, because they've worked so hard to get up there, they literally have nothing left. Which is why it's so wonderful. You can go to the top of a river, and then you just go with a net and you scoop up all these salmon that have already laid their eggs. And you're like, they're going to die anyways, but they still are so delicious, except for they've used up a lot of their fat reserves, so it's not as fatty as salmon usually is. But anyways, it's just a little flakier, you know? Y'all don't eat fish? Wrap it in bacon, and then, you know. All right, so we can pump materials against the concentration gradient, and this is called active transport. This requires energy. Every molecule you pump requires energy. Every one, because you're forcing it to go the opposite direction from where it wants to go. You have to remember to think about, you need to associate concentration gradient with potential energy. Because if you remember back to your physical science class, what is the textbook example of potential energy? A rock sitting on top of a cliff, right? Ready to fall? That's like the textbook example of potential energy. How do you get that rock up there? You push it up the wall, right? Or you push it up the thing. Who's that Greek guy that had to do that for all of eternity? That just kept pushing the boulder up the hill only to have it fall back down? Wait, what's that dude's name? I don't even remember. Anyways, Prometheus? No, it wasn't Prometheus. Prometheus, I think, is Roman, isn't he? Is Prometheus Greek? I don't know. All right. So membranes are selectively permeable. We talked about this. Polar molecules move very slowly through membranes. Charged atoms and molecules are completely blocked. Meaning if these need to move, you have to have a protein to facilitate it. These, they can probably move through, but it's not going to be very quickly. So if you want a lot of them to move through, you need a protein to facilitate it. All right? Does that make sense? It's awesome, right? You're like, I don't know, I stopped listening after you said duck bacon because now I want to go hunting. Is it duck season? I don't think it is. All right. So here's a nice chart. This is a chart. It's not out of your textbook. I borrowed it from another one where it talks about the nice difference between simple and facilitated, but still passive, not requiring energy, and active transport where you're pumping materials against their concentration gradients. So this is a good one. Here's the completion of this. And so this chart, it's going to be a healthy, it's a healthy table to know. I don't know why I keep stuttering. I'm so excited. But it's a good table to familiarize yourself with. All right. It is presently 12:40, which is the time that our class ends. And so, unfortunately, I'm going to have to say goodbye to you and wish you a very nice weekend. I mean, even a nice conclusion to your Friday. That's not the unfortunate. The fortunate thing is I have to say goodbye. I'm sorry.